So another major part of chapter 2 is how proteins are created really beginning in the nucleus of the cell. And this is a process called transcription and then translation of proteins. When you think about transcribing something, it means really to write something down, to write it down. And so what's being transcribed is a gene, which is basically it's a gene on the DNA strand and nucleus of the cell. And a gene codes or essentially is the how exactly to build the primary structure of the protein. So basically, it's the recipe for a protein. And so transcription is basically writing down that recipe because you can't take the DNA part, the gene, out of the nucleus. So you have to write down the recipe for a protein. And that written form is called a messenger RNA. Whoops, why did I put S? So messenger RNA is transcribed off a gene on the strand of DNA in the nucleus. And then that messenger RNA travels out to the cytosol in the cell and is translated. What does translated mean? Well, it's taken from one language, which is a messenger RNA, and it's converted into a primary protein structure as the messenger RNA is read. And so that's what translation is. And so this is going to be a kind of a quick overview of transcription and translation of proteins. And again, remember, proteins are involved in all physiological processes. So it can be cellular signaling like hormones or neurotransmitters. It could be tissue remodeling, right? Actin and myosin strands in muscle cells. It could be functional. It could be enzymes, right? And so lots of different types of proteins that are doing different functions. And the DNA found in the nucleus of the cell is what directs the beginning of the synthesis of a protein and also transcribes how to assemble that protein in the cell. So transcription is basically taking a gene, one part of the DNA strand, which basically is a recipe for one protein. So one gene is going to code for one protein, and it's being transcribed onto a messenger RNA. And that messenger RNA is the transcribed gene. It's going to leave the nucleus of the cell, go out into the cytosol of the cell, and be translated into a protein, right? And so that translation is reading messenger RNA and finding correct amino acid to build primary protein structure. Okay, so DNA is in the nucleus of the cell. So this is the cell, right? Here's a cell, and this is the nucleus. And you have DNA, and it contains genes. And these genes contain information, information specifying amino acid sequences and proteins. Transcription is getting the genetic information off the DNA strand and transcribing it to a messenger RNA. So that's an mRNA. And this occurs in the nucleus of the cell. And that messenger RNA is going to leave the nucleus via a nuclear pore and go out into the cytoplasm. Cytoplasm is both organelles and cytosol. So it's out in the cytosol. And that messenger RNA strand is going to be translated with the help of ribosomes. And those ribosomes are going to connect the amino acids with peptide bonds and create a primary structure of a protein. And then that primary structure will fold a second time due to hydrogen bonding, secondary structure, and then the tertiary structure, and it'll be finalized, right? So translation is a process that uses coded information in the messenger RNA to assemble the protein. And this occurs in the cytoplasm, specifically the cytosol. And so the genetic information flow is from the DNA and creating a messenger RNA off that DNA. And from the messenger RNA, it's going to be translated into a protein. And so again, this is transcribing a messenger RNA, I'm sorry, DNA into a messenger RNA. And then messenger RNA goes out into the cytoplasm and translates its code into a protein with the help of ribosomes, which put the amino acids in the correct sequence. And this forms the primary protein structure. So genes, definition, it's a portion of the DNA that holds the, quote, genetic code, or I call it the recipe for a specific protein. It's a sequence of DNA nucleotides that contain information that specifies which amino acid goes next to which amino acid. Now we talk about the genome, like the human genome. That's all the genes in the DNA, right? And that's going to be all of the different genes. So this is showing you one gene, one protein, codes for one protein. And each triplet on a gene, on the DNA strand, is coding for an amino acid. And so this is one amino acid, there's another amino acid, another amino acid. And so during transcription, there's the formation of a messenger RNA. Messenger RNA. And so it's basically writing down each triplet. And so on the messenger RNA, the triplet is called a codon. Because it's not exactly a triplet, right? G corresponds to C, T corresponds to A, C corresponds to G. You don't have to know this for this class. We're not microbiology. But understand that each triplet is coding for a single amino acid, and that codons are basically what is going to also denote the same amino acid on a messenger RNA. So it's just writing down each amino acid. And so steps of protein synthesis, again, for review, DNA holds the code for each individual protein. That's the genetic code. It is transcribed or written down, which amino acids going next to which amino acids, from a triplet on a DNA strand to a codon on the messenger RNA, right? So you have triplets that get transcribed into codons on the messenger RNA strand. And then the messenger RNA strand is going to move out of the nuclear pore into the cytoplasm, and it's going to then be translated with the help of ribosomes in the cytosol. So this is, again, just reviewing. DNA strand contains the code for the triplets. It's called the template strand. A triplet codes for a single amino acid. Triplets have a complementary 3 base sequence in a messenger RNA, and that's called the codon. So codon is essentially a triplet on a messenger RNA strand. And a codon, they code for the same sequence of amino acids as the triplets. And this is what transcription is. It's being transcribed or written down onto a strand that can leave the nucleus of the cell, because DNA strands can't leave the nucleus of the cell, but messenger RNA can. And so each codon is going to code for one amino acid. There's also an initiation codon and there's a termination codon. So it says we're going to start, the gene starts here and the gene ends here. And so those triplets say start, stop. There's 3 types of RNAs that are transcribed. We've been talking about messenger RNA because messenger RNA have the codons for building a protein. Ribosomal RNA help read messenger RNA and assemble the amino acids and create peptide bonds. Transfer RNA, tRNA, they receive info on what amino acid to bring to ribosome messenger RNA complex. So these guys are the scavengers. They're going around, tRNAs are going around and finding the correct amino acids that they're told to bring over to the chain. Now, RNA polymerase, ASE, this means it's an enzyme. And it's an enzyme that oversees, the synthesis of messenger RNA. It helps unwind the DNA template. It adds complementary RNA nucleotides and catalyzes the formation of bonds between the codons to create a messenger strand. And then it stops when the termination signal, it arrives at the termination signal. So again, the 3 types of RNA. So you've got messenger RNA, carrying the instructions on how to build a protein in the cytoplasm. You have ribosomal RNA. It's going to help translate or read the codons on the messenger RNA and assemble amino acids and form peptide bonds between them. And then transfer RNAs. Now, these are going to bind to the amino acids in the cytoplasm and bring it to the ribosomal complex, and then it's going to begin the process of attaching. And then that tRNA will release the amino acid and then eventually go find the next amino acid. And so transcription of DNA to messenger RNA, you have the initiation codon it's going to then, with the help of RNA polymerase, unwind the strand where that gene is. You're going to have a triplet here helping to form codons for each amino acid, and this is going to become the messenger RNA. When you see a stop codon, that RNA polymerase is going to help separate, and the messenger RNA then can go out to the cytoplasm and be translated. So again, definition. Transcription. DNA gene base sequence is creating a complementary messenger RNA base sequence. Basically, it copies the information of the exact order of amino acids needed to make a specific protein. Transcription factors, they act as gene switches to turn on and off. Basically, the process of building proteins. So it's the switch to start building proteins in the cell. And it can become active or inactive, depending on methylation and different factors. Things can slow down or speed up the transcription of proteins. So what this is, factors that affect, building of proteins. So think cellular metabolism. You need enzymes to transfer energy from the food you ate. Basically, all the food that you're eating, you need to transfer the energy in that food to ATP molecules. So you're using the energy that you consumed. But you need enzymes to make that happen. And enzymes are proteins. And if you don't have enough of those enzymes, then you're not going to transfer enough energy, and you're just going to store that energy as fat. Or hormones are also proteins structures. And so if you don't have enough of those hormones being built, this podcast right here, if you touch that and you open it, it's going to take you to a podcast. It's about 10 minutes long on studies about exercise and basically turning on transcription factors to build more enzymes, metabolic enzymes, and proteins. So listen to that. It's your discussion podcast in this module. So with the messenger RNA transcription, you're going to have initiation where the RNA polymerase starts, unwinds the DNA, and it's going to start reading the triplets and creating codons and building a messenger RNA strand, and that is called elongation. It's going to get to the stop codon, and that's going to be termination. It's going to release the messenger RNA off the DNA strand so it can wind back up. So initiation with help of transcription factors, it's going to pry apart the DNA strands and initiate the messenger RNA, and it's going to elongate with each triplet being written down into a codon on the messenger strand. And when it gets to the stop code, it's going to terminate. So this kind of goes over a review. And I like myself, when I'm trying to learn this stuff, I always go, what happens first? Well, RNA polymerase is binds to a promoter on the template strand. That's going to then separate the two DNA strands. And then you're going to have elongation. The RNA polymerase moves along the template strand, reading each triplet and creating a codon. Once that happens, upon reaching a stop signal, RNA polymerase releases the newly made messenger RNA. There's something called spliceosomes. I'm not going to test you on this, but it basically kind of cleans up the messenger RNA. So you have a now formed messenger RNA that can go out into the cytoplasm to then be translated. Quick word here. And again, you don't have to memorize all these things, but it's important to understand that the triplets code for, and the triplets on a DNA strand are, you're going to have a codon formed on a messenger RNA. Well, each of those codons, you can have multiple codons code for the same amino acid. So these two are going to code for this amino acid. These two are going to code for this. Some, like leucine, have 6 different codons that code for it. So it gets really complicated. Hopefully you guys will get to microbiology and know a little bit more when this gets brought up, because you get to learn all about this wonderful stuff. But for our purposes, it's an FYI. Translation. Messenger RNA attaches to a small ribosomal subunit, and then a second larger ribosomal subunit is going to attach on the other side of the messenger RNA strand. So it looks like this. This would be the messenger RNA strand. And so this is the large ribosome, and this is the small. And these yellow beads right here each represent an amino acid. And so an anticodon, or a version or image of the codon, is put onto a transfer RNA. And that transfer RNA is going to find the amino acid, bring that amino acid to the protein chain. And new amino acids are added by other transfer RNAs as the ribosome moves along, forms peptide bonds until it reaches the stop codon on the messenger RNA. And so this is kind of showing you that these are string of amino acids read off the messenger RNA, right? So this would be the primary structure of a protein right there. This is what a transfer RNA is. If it's charged, it means it has an amino acid bound to it. If it's free, it has no amino acid bound to it. Again, it's reading the codon off the messenger RNA, and that's going to code for a specific type of amino acid, and it's going to go find that amino acid, and it's going to bring it to the complex. So this is kind of a cute version of it, right? So this right here is the messenger RNA strand. This is the small ribosomal subunit. This is the large ribosomal subunit. These right here are the tRNAs. And these two tRNAs, this is the codon on the mRNA, and this is the anticodon that codes for that amino acid. This is a charged tRNA. And it's going to go find that amino acid that it's called for, and it's going to bring it over to the ribosomal subunit. So here comes the tRNA with the next amino acid. It's going to bring it over, right? And a peptide bond is going to be formed here once it's brought over. And as that peptide bond forms, you're going to now have a free tRNA to go again, come back and read the next codon. And so you're going to have this protein with each tRNA coming over, bringing it, new peptide bond forms, new tRNA leaves, and the tRNA is released from the site, and a new tRNA is going to bind, and that's how we form the primary structure of a protein. I'm running out of room. Oh, so I'll just write it down here. Primary structure of protein. And that is translation in cytoplasm. Getting sloppy now. What happens to these primary structures of the proteins? Well, there is something called a leader sequence, and that tells the cell what is supposed to happen to that particular protein. And so the leader sequence gets translated, and these leader sequences can say, hey, it's going to hang out in the cytosol, or it's going to go to the endoplasmic reticulum to finish forming, formation. If there's no leader sequence, it just stays in the cytosol. So this is kind of showing you the leader sequence. Move to the nucleus. It can stay in the cytosol. It can move to a peroxisome. It can move on to mitochondria, right? It's an enzyme that would move to the mitochondria. Two more things. Copying DNA is called replication. Chromosomes. Chromosome is one complete DNA molecule plus associated proteins. So humans have 23 pairs of chromosomes. I should say humans donate 23 pairs of chromosomes. You're going to get one set from your father, one set from your mother, and all 23 chromosomal pairs are going to make up a human genome, right? So they're pairs, so there's really 46 total. But you're getting 123 set from mom, 123 set from dad, and you end up with 46, right? Or 23 pairs. When a gene codes for a wrong amino acid, or it changes the sequence of the primary structure, it changes the amino acids in the sequence, that's called a mutation. And different things can cause mutations. It can be radiation, it could be chemicals, it could be just natural selection. Some of these changes are good changes and can change the structure of a protein to be more efficient or effective. They could be bad changes and render a specific enzyme disabled, and therefore you can't, digest milk sugar. I mean, there's just variations, but these are what mutations are. All right, on to the next, which will be protein conformation.